Welcome to The Explainer. Today, we're tackling what seems like an impossible biological puzzle. How does every single one of our cells manage to solve this incredible packing problem, squeezing a massive amount of information into a space that's literally smaller than a speck of dust? It all kicks off with this number, 2 meters. That's about 6.5 feet. Now, just try to picture taking a thread that long and stuffing it into something so microscopic you can't even see it. And here is where it gets kind of mind-bending. Every one of your trillions of cells has one of these 2-meter-long DNA strands, but it has to fit inside a nucleus that's only 5 to 8 micrometers wide. To give you some perspective, that's like trying to cram a thread over 20 miles long into a single tennis ball.
So you gotta ask, how does it all fit? I mean, it seems impossible. If you just tried to jam it all in there, the cell would literally burst. But, of course, nature has this beautiful, elegant solution.
Okay, so let's back up. What is this thread we're trying to pack? Well, it's DNA, deoxyribonucleic acid, the famous blueprint of life. Every single instruction for building and running your entire body is written right into this one molecule.
Now, we all know DNA for its iconic double helix shape. And yeah, it's incredibly thin, just 2 nanometers wide. But you know what? For our packing problem, the most important property isn't its shape or its size. Nope, the real secret comes down to something you can't even see. It's electrical charge.
And this is the key, right here. The backbone of that long, spiraling DNA strand is negatively charged. It's basically a super long, negatively charged string. And that one simple fact, well, that's the secret to solving the whole problem.
Okay, so if you've got this long negative thread, how do you control it? Nature has the perfect tool for the job, a group of proteins called histones. Think of them like tiny little spools for our genetic thread.
And this is where the genius comes in. Histones are positively charged. You see where this is going, right? Negative DNA, positive histones. It's basic physics. Opposites attract. And that simple, fundamental force is the engine that powers this entire packing process.
The whole thing works in these really neat layers. First, that negative DNA strand wraps itself super tightly around those positive histone spools. This creates these little individual units, which then link up, kind of like beads on a string. And then, finally, that whole string of beads gets coiled up again, creating an even thicker, more compact fiber.
So that little bead on a string unit, it actually has a name. It's called a nucleosome. It's just a piece of DNA wrapped around a core of those histone proteins. This is the first and arguably the most crucial level of packing everything down. All right, so we've got our beads on a string, but the packing doesn't stop there. These nucleosomes are then coiled and stacked together, forming a thick, dense fiber called chromatin. This is the final state. This is how DNA actually exists inside the nucleus, all packed up and ready to go. And just look at the difference all that coiling makes. We've taken a flimsy 2-nanometer DNA strand and turned it into a robust 30-nanometer chromatin fiber. That's a 15-fold increase in thickness, which is a huge leap in compacting all that information. Now, you might be thinking this all sounds like a tangled-up mess, but it's not at all. It's actually more like a perfectly organized library. This packed chromatin has specific landmarks, so the cell knows exactly where to find anything it needs.
Yeah, it's pretty incredible. Even when it's coiled up this tight, the cell knows the exact address for every single gene. That's the gene locus. And there are other specialized regions, like the centromere, a super-condensed area, and the kinetochore, which basically acts as a handle for the cell's machinery to grab onto during division.
Okay, but all of this is happening at a scale that's just mind-bogglingly small. So it makes you wonder, right? How in the world do scientists actually see any of this stuff? It's not like you can just use a regular microscope. Well, they use a pretty clever trick. Scientists use special chemical stains, like methylene blue, which are designed to bind directly to the DNA molecule. This interaction basically makes the chromatin light up, so we can finally see these thread-like structures with a powerful microscope. But there's a big catch. To get these incredible images, the staining process actually kills the cell. So what we're seeing is a perfect frozen-in-time snapshot of this structure. We can't actually watch the living, dynamic process in action.
So when you just step back and take it all in, from a 2-meter-long molecule to a structure that fits inside a space just a few micrometers wide, you realize this is an absolutely magnificent feat of biological engineering. It's a truly awe-inspiring system of organization, designed for one purpose: protection.
And that leaves us with one final, pretty profound thought. Nature went to all this incredible trouble, using physics, creating spools and coils and supercoils to build a system of near-perfect biological storage. It really makes you stop and ask, what does it say about the information inside, the very blueprint for life, that it is worth protecting with such impossible elegance?
You know, have you ever stopped to think about the instruction manual that's packed inside every single one of your cells? Well, today, we're going to dive into that incredible genetic blueprint and see how it tells its story. So let's start with a classic example, something a lot of us can do: rolling your tongue. Whether you can do it or not is literally written into your biological code. But how does that even work? Where is that little piece of information actually stored? Get this. The instruction for that one simple trait is written on a molecule that, if you were to stretch it out from just one of your cells, would be about 2 meters long. I mean, that's taller than most people.
So that presents a pretty mind-boggling packaging problem, right? How on earth does your body cram 2 meters of this stuff into a microscopic cell nucleus that's only about 5 to 8 micrometers wide? To put that into perspective, it's like trying to stuff 40 kilometers of superfine thread into a tennis ball. It seems impossible.
Well, the solution is just a masterclass in organization. The DNA strand wraps itself tightly around these little proteins called histones. This creates a structure that looks a lot like beads on a string. Then, that string of beads, called nucleosomes, coils up on itself again and again, first forming something called chromatin, and then it gets even more condensed into the structures we can actually see.
And this final, super compacted form of your DNA, that's what we call a chromosome. You can think of it as the ultimate biological hard drive, the perfect storage solution for your entire genetic library.
Okay, so how is this library actually organized? Well, inside pretty much every single one of your cells, you've got 46 of these chromosomes, and they're all neatly arranged into 23 pairs. These pairs have a special name: homologous chromosomes. Now, homologous is just a fancy way of saying they match. They're the same size, the same shape, and, most importantly, they carry the instructions, the genes, for the same traits in the exact same order. And this is where your personal story really begins. See, in each of those 23 pairs, one chromosome comes from your mother, and the other comes from your father. They line up perfectly. Same size, same shape, same list of genes. But notice that last row? The specific versions of those genes, which we call alleles, they aren't necessarily the same. And that's a huge deal. So this brings up a really important question. If they're a matching pair, does that mean they're just exact, identical copies of each other? And the answer is a big, resounding no. Absolutely not. And this one fact is the key to understanding all the genetic diversity that makes you, well, you. The reason they aren't identical comes down to this idea of alleles. Alleles are just different versions or flavors of the same gene. Think of it like this. The gene is for eye color, but the alleles are the specific instructions for blue eyes or brown eyes or green eyes. Same gene, different outcomes. Let's circle back to our tongue rolling example. The gene for that trait sits at a very specific spot on a chromosome. But you can have different alleles there. One version, let's call it big A, gives the instruction, yes, you can roll your tongue. The other version, little a, gives the instruction, nope, can't roll it. Now, since you get one of these homologous chromosomes from each parent, you get one allele from your mom and one from your dad. So your mom might give you the big A allele while your dad gives you the little a allele. And that specific combination of alleles you end up with, that's what we call your genotype. So for tongue rolling, your genotype might be big A, little a. If we were to look at another trait at the same time, like say eyelid type, maybe big B, little b, we'd write out your genotype just like this. It's your unique genetic recipe. Okay, so that explains the relationship between genes on matching chromosomes. But what about totally different genes that just happen to be physically located on the same chromosome, one right after the other? We call those linked genes. And it kind of makes sense, right? Because they're physically part of the same long strand of DNA, they usually get inherited as a package deal. Now, it's really important to remember that even though these genes are linked, they are non-homologous. That just means there are instructions for completely different things, like one might be for tongue rolling, and its neighbor could be for something totally unrelated.
So let's tie this all together real quick. Your unique identity starts with this incredible packing job, 2 meters of DNA squeezed into 46 chromosomes. Those chromosomes come in 23 homologous pairs, one set from each parent. And while the pairs carry the same genes, it's the different versions, the alleles, that you get from your parents that create your one-of-a-kind genotype.
It's really amazing, isn't it? This elegant system of pairing, variation, and combination is the engine for all human diversity. It just makes you wonder, what other secrets about who we are hiding in plain sight, just waiting for us to unlock them in our own genetic code?
Welcome to The Explainer. Today, we're going to decode the simple counting system that acts as your entire genetic blueprint. Yeah, it's the biological math that makes you, well, you. And you'll see that when this count is off by even 1, the consequences can be huge.
So, let's kick things off with a bit of a riddle. This might seem random, but the answer actually gets to the heart of a fundamental rule for how your DNA is organized. And that rule is all about one simple concept.
And that concept is the set. Before we can even think about counting chromosomes, we got to get on the same page about what makes one complete functional set.
Okay, so think about it like this. You're ordering a combo meal at a fast food place. You get 1 burger, 1 order of fries, and 1 drink. That whole package, 1 of each thing, that's one set, a complete functional meal.
Right. So following that logic, if you bought 2 burgers, 2 fries, and 2 drinks, you don't just have a random pile of food, right? You've got 2 complete sets, 2 full meals. Simple enough? Now let's see how this applies to our biology.
Okay, so this is where it gets really cool. Most of the cells in your body are what scientists call diploid. And all that fancy word means is that they carry 2 complete sets of genetic instructions, basically 2 full sets of chromosomes. You get 1 set from your mom and the other from your dad.
Now, biologists have this really neat shorthand to describe these sets. And trust me, once you get the hang of it, you'll be able to look at pretty much any cell and figure out its genetic makeup in a snap.
For us humans, the magic number is 23. That means one complete set of our genetic instructions is made up of 23 unique chromosomes. You can think of them as 23 different types of instruction manuals.
This brings us to the famous formula for humans, 2n equals 46. So what does that mean? Well, that letter n stands for the number of unique chromosomes in one set. Since n is 23 for us, and most of our cells are diploid, or 2n, you just do the math. 2 times 23 gives you a grand total of 46 chromosomes.
But here's something really interesting. That n number isn't a universal constant. A tiny fruit fly, its n is only 4. A chimpanzee, our close relative, has an n of 24. And that just goes to show you, having more chromosomes doesn't necessarily mean an organism is more complex. It's all about what's in those instructions.
Now, a quick side note. You might also bump into the letter A when people talk about this. Technically, N is for chromatin sets and A is for DNA sets. But for what we're talking about today, the number is the same. So don't let it trip you up. If you see 2N or 2A, just think, okay, that means 2 complete sets. All right. You ready to put this to the test? Let's try a couple of quick puzzles. This is pretty much exactly how biologists figure this stuff out.
Okay, so take a look here. We've got a cell with 3 long chromosomes and 3 short chromosomes. So using our new N and A language, how would you describe this?
Okay, here's how you solve it. First, you have to count the number of types of chromosomes. In this case, we've got 2 types, right? Long ones and short ones. So that means our N equals 2. And since we have 3 of each type, that means we have 3 complete sets. So we'd call this cell 3N or 3A. Simple as that.
Okay, let's do one more just to make sure it sticks. This cell has 2 long, 2 medium, and 2 short chromosomes. What's the breakdown here?
So, same process. First, count the types. We've got long, medium, and short. That's 3 different types, so N equals 3. And since there are 2 of each kind, we have 2 complete sets. That makes it a diploid, or 2N cell. You see the pattern now, right?
So it's pretty clear this counting system is super important for life to work properly. Which begs the question, what happens when a cell gets the count wrong?
Well, sometimes, during that incredibly complex dance of cell division, a mistake happens. An error occurs, and the cell can end up with one extra chromosome. It's a situation we describe as 2n plus 1.
And there's a formal scientific name for this. When a cell has an abnormal number of chromosomes, either one too many or one too few, it's called aneuploid.
These simple miscounts, these aneuploidies, they're actually the genetic reason behind several well-known conditions in people.
For example, Down syndrome, which you might also hear called trisomy 21, is a textbook case of that 2n plus 1 error. It means that instead of the usual 2 copies of chromosome 21, an individual has 3.
And it's not just a numbered chromosomes. These counting errors can happen with our sex chromosomes too. This leads to conditions like XY syndrome, triple X syndrome, or Turner syndrome, where a female has just a single X chromosome instead of 2. These are all different forms of aneuploidy.
So you can see this isn't just some academic exercise in counting. Getting this number exactly right is absolutely fundamental. From the way our cells work to how our entire body develops, everything relies on this perfect account.
And that really leaves us with something pretty profound to think about. This delicate, almost mathematical balance is literally the foundation of who we are. And the fact that it's so fragile, well, it's a powerful reminder that all of life, in all its mind-boggling complexity, is really built on a set of rules that are, in a way, beautifully simple. Thanks for joining me.
Welcome to The Explainer. Today, we're diving into a really fascinating biological construction project. How a plant cell, of all things, manages to divide itself into 2.
So think about this. Unlike an animal cell, a plant cell is stuck inside this tough, rigid box, its cell wall. And that presents a pretty big problem. How in the world do you split yourself in 2 when you're trapped inside a solid container?
Well, animal cells, they kind of have it easy. They just pinch in the middle, form what we call a cleavage furrow, and then pop, 2 separate cells. But plant cells, they can't do that. They have to get way more creative. They literally build a brand new wall right down the center, from the inside out. Okay, let's get into how they pull off this incredible feat of engineering.
All right, first, let's look at the master plan. Plant cells are actually missing a key piece of equipment that animal cells use for division, something called centrioles. So how do they even begin to organize this whole complex process? It turns out they use something called microtubule organizing centers, or just MTOCs for short. You can think of these as 2 command centers, or poles, set up at opposite ends of the cell. They marshal all the forces, they assemble the protein tracks needed to pull everything into place, and they prove you don't even need centrioles to get the job done.
So, with our command centers in place, it is time to start the actual construction. And the very first step is to bring in the building materials for that new wall. The cell's Golgi apparatus, which is kind of like a factory and a post office all in one, starts shipping out these tiny little bubbles called vesicles. And they're not empty. Nope, they are packed with pectin, which is this really sticky substance that's going to act like the mortar for our new wall. And here is where we really get to see that inside-out strategy in action. These Golgi vesicles, they don't start at the edges. They travel along these protein tracks right to the cell's equator. And there they all line up and start to merge together, forming a new, still pretty fragile structure right in the middle of everything.
And this initial structure has a name. It's called the cell plate. It's really just a collection of all those tiny pectin-filled bubbles, all lined up and ready to become a permanent wall. It's the foundation for the partition that's about to separate the 2 new cells. But you know, a fragile plate of bubbles is not going to cut it. It's not strong enough to divide a cell. It needs to mature into a solid, permanent partition. So how does the cell finish the job? Well, that cell plate starts growing outwards, expanding like a disc until it connects with the original cell wall on all sides. As it does that, the membranes of all those little vesicles just seamlessly merge to become brand new cell membranes for the 2 daughter cells. And at the same time, all that sticky pectin they were carrying gets released into the space between them. This new layer of pectin is called the middle lamella, and its job is basically to act like cement,
gluing the two new cells firmly together. Once that glue is in place, each new cell gets to work building its very own primary wall right alongside it. And just like that, the division is complete. But this unique inside-out construction method leaves behind this fascinating and absolutely vital legacy. You see, when all those vesicles fuse together, the process isn't always 100% perfect. Tiny little gaps or channels are left behind in that new wall, and they create these direct passages between the two neighboring cells. These incredible little intercellular bridges are called plasmodesmata. And what these channels do is just amazing. They create a direct highway so cells can share nutrients and send signals back and forth. But this connection has a downside. The highway is so efficient that it also gives plant viruses a perfect route to spread really quickly from cell to cell throughout the entire plant. This huge interconnected network of shared cytoplasm is called the symplast. And what it means is that, in a very real sense, a plant isn't just a collection of individual walled-off cells. It's a unified, communicating superstructure. And it's all thanks to these tiny passages that were left over from cell division. So the really amazing thing to think about is that the very method a plant cell uses to divide, building that wall from the inside out, is what fundamentally defines how the entire plant lives and communicates. And that leaves us with a pretty cool, provocative thought. If their cells are all this deeply interconnected, should we be thinking of a plant less as a colony of individuals and more as one single, sprawling, continuous superorganism? Welcome to The Explainer. Today, we're jumping into the grand finale of one of biology's most important processes. We are talking about meiosis II, the final, crucial step that makes sexual reproduction, well, possible. So let's think about this for a second. It's a fundamental challenge that all life has to solve. How do you create the next generation without just doubling your chromosomes every single time? I mean, that would get out of hand pretty fast, right? Well, nature came up with this really elegant two-part answer called meiosis. The entire goal here is to get from what we call diploid to haploid. You can think of it like this. A normal body cell, a diploid cell, has a complete two-volume encyclopedia of your genetic code. But a gamete, a reproductive cell, only gets one of those volumes. This is key because it means when two gametes come together, the new organism gets a full set, one volume from each parent. Perfect. Now, this whole thing doesn't just happen all at once. It's kind of like a two-act play. And to really get what's happening in Act 2, we've got to do a quick recap of Act 1, meiosis I, the first major division. Okay, I know what you're thinking. This notation looks like some kind of complicated algebra. But trust me, it's actually just a simple way to keep score of our genes. So N is the number of chromosome sets, and A is the amount of DNA. A cell copies its DNA right before this all starts, so it becomes 2N4A, 2 sets of chromosomes, 4 times the base amount of DNA. Now, the big job of meiosis I is to separate the homologous pairs, that is, splitting up the chromosome you got from your mom and the one from your dad. After that first division, we have 2 cells that are 1 and 2A. We've halved the sets, but look, each chromosome is still a duplicated copy, so the job's not quite finished yet. And that brings us to the main event, meiosis II. This is where the cell gets down to business and makes that final precision cut to create a true functional gamete. You know, at first glance, meiosis II looks a whole lot like regular cell division, or mitosis. But there's a huge difference. The cells going into this stage are already haploid. So here's what happens. The machinery for division gets set up again, that molecular glue holding the copied chromosomes together finally dissolves, and then they get pulled apart to opposite ends of the cell. And this is the pivotal moment. Sister chromatid separation. Those identical, duplicated copies of each chromosome that have been joined this whole time, they're finally separated. This single action is what turns a duplicated chromosome into 2 individual ones. And that's what makes a true gamete possible. It's the whole point. So, after those 2N2 cells from meiosis I both go through this final division, what are we left with? We started this whole journey with just 1 cell. And after these 2 intense rounds of division, we end up with 4. 4 brand new cells, and each one has a totally unique genetic combination. And here is their final genetic score, 1N1A.
What does that mean? It means each of these 4 cells has just one set of chromosomes, and each of those chromosomes is made of a single strand of DNA. They are officially haploid gametes, ready to go. Mission accomplished.
Phew! We've gone through a lot of stages and codes, so let's zoom out for a second and just trace the entire journey of our genetic material from the very beginning to the very end.
This right here is the whole story in one shot. We start with a regular diploid cell, 2N2A. It prepares for division by copying its DNA, becoming 2N4A. Meiosis I does the first big reduction, giving us 2 1N2A cells. And then meiosis II performs that final precise split, leaving us with our 4 1N1A products. It's just a beautiful 4-step cascade of division.
So let's just boil it all down. Meiosis is a 2-act play, each with a different job. Meiosis I is all about separating the chromosome pairs you inherited from your parents. But meiosis II, the star of our show today, is about separating the copies of each of those chromosomes. And the final result? 4 unique haploid gametes. It's this incredible process that keeps the number of chromosomes stable generation after generation. This whole thing is like a perfectly choreographed cellular dance, and it's absolutely essential for life as we know it. But because it's so complex, it's not always foolproof. So what happens when this intricate dance has a misstep? Well, the consequences can change a life before it even begins. And that is a story for another explainer.
You know, every single day, your body is doing something incredible, something you don't even notice. It's creating trillions of brand new cells for growth, for repair, you name it. But have you ever wondered how one tiny cell perfectly becomes 2? Well, today, we're going to dive into the elegant and super intricate biological dance that makes it all possible, mitosis. So before a cell can even think about dividing, it has to solve this massive packaging problem. I mean, the genetic blueprint for you, your DNA, is unbelievably long.
That's right. 2 meters. If you could take all the DNA from just one of your cells and stretch it out end to end, it would be taller than most people. And all of that has to get crammed into the cell's nucleus, which is only about 5 to 8 micrometers across. To put that in perspective, it's like trying to fit a 40-kilometer-long piece of thread into a tennis ball. So how on earth does the cell pull that off? Well, the solution is just some brilliant chemical engineering. The cell uses these special proteins called histones. You see, DNA has a negative charge, and these histone proteins, they have a positive charge. And, as we all know, opposites attract. This super simple attraction is the secret to the entire packaging process.
So the long, negatively charged DNA strand just wraps itself tightly around these little positively charged histone spools. This little bundle is called a nucleosome. Think of it like thread on a spool. Exactly. Then these nucleosomes start coiling up and stacking together, condensing everything into a much more manageable fiber we call chromatin. For most of its life, this is how your DNA is stored. All right, now that all the genetic material is packed up neatly, the cell can get ready to divide. But, and this is a big but, before it can split, it has to make a perfect, identical copy of its entire DNA blueprint. This super important step happens during a time called the S phase. So during this S phase, something pretty amazing happens. Every single strand of that chromatin gets perfectly replicated. Now you have the original strand and its brand new copy, which we call sister chromatids. They're held together so tightly that if you look under a microscope, they still look like one single thread. At the exact same time, the cell's little organization hubs, the centrosomes, they also make a copy of themselves. From these new centrosomes, you start to see these star-like fibers called asters radiating out. This is all about setting the stage for the big split. Okay, so we've got 2 complete sets of DNA and 2 organizing centers. The cell is officially ready for the main event, mitosis itself. And honestly, the best way to think about this is like a perfectly choreographed dance in 4 acts. It's all about making sure each new cell gets exactly 1 full copy of the genome. Our little cellular performance unfolds in 4 distinct acts. We've got prophase, metaphase, anaphase, and telophase. Let's take a look at each step of this choreography. The dance kicks off with prophase. This is where that chromatin condenses like crazy. We're talking over a 10,000-fold coil up. It gets so thick, about 1,400 nanometers wide,
that for the first time, we can actually see these distinct thread-like chromosomes with a regular light microscope. Now, to let all the machinery get in there, the membrane around the nucleus, it just disappears. And these molecular ropes, called spindle fibers, start to form and reach out for the chromosomes. Next on the dance card is metaphase. This is a moment of perfect, beautiful order. Those spindle fibers, coming from opposite ends of the cell, start this little tug of war. They pull and they push the chromosomes until every single one is lined up perfectly in a single file line right down the middle of the cell. We call this imaginary line the metaphase plate, or equatorial plate. And this alignment, it's an absolutely critical checkpoint. It's the cell's way of doing a head count, making sure that when it finally splits, each new daughter cell is going to get one copy of every single chromosome. No more, no less. So once everything is lined up just right and the cell gets the all-clear signal, we move on to the most dramatic, action-packed part of this whole dance. Boom! This is anaphase. The proteins holding those sister chromatids together just let go. The sisters are ripped apart, and each one is now considered a full-fledged chromosome. The spindle fibers start shortening, yanking the chromosomes toward opposite ends of the cell. And they're pulled so hard and so fast that they actually get dragged into this characteristic V shape. It's pretty cool to see. Okay, so this is a really important detail to get right. Before anaphase, we've got one chromosome that's made of two sister chromatids, right? But the second they separate in anaphase, we now have two totally independent chromosomes. So for just a brief moment, the cell actually has double the normal number of chromosomes. The final act of this nuclear dance is telophase. The newly separated chromosomes have finished their journey. They're at opposite poles of the cell. And now, the cell basically just hits the rewind button on everything it did back in prophase. The chromosomes start to uncoil and relax, going back into their loosey-goosey chromatin form. A brand new nuclear membrane forms around each complete set, creating two new totally identical nuclei. The spindle fibers, their job is done, so they just break down and disappear. And with that, the mitotic dance is over. But hang on, we're not quite finished. We've successfully made two nuclei inside one cell, but now we gotta divide the rest of the cell, all the goo, the organelles, everything. We need to split it into two separate daughter cells. And this process is called cytokinesis. And how this happens really depends on what kind of cell we're talking about. Animal cells, they've got a flexible membrane, so they form what's called a cleavage furrow. A ring of proteins tightens around the middle like a drawstring, pinching the cell until it just pops into two. But plant cells, with their stiff cell walls, can't do that. Instead, they build a new wall called a cell plate right down the middle, and it grows outward until it creates a solid partition. So, why do we just spend all this time watching this tiny, microscopic dance? Well, because this precise, carefully controlled process is, without a doubt, fundamental to who you are and life itself. Seriously, every time you heal from a cut, every single moment that your body is replacing old red blood cells, and yeah, the entire journey of growing from one single fertilized egg into the person listening to this right now, it's all thanks to mitosis. It is the engine of our growth and our regeneration. And here's a final thought for you. This incredibly complex dance happens with almost perfect accuracy, trillions of times a day, all over your body. But what happens when a cell messes up? When it misses a step in the choreography? What are the consequences when this beautifully controlled process goes wrong? Now that is a question with some pretty profound implications for our health, and definitely something to think about. All right, let's dive into one of the most amazing processes in all of biology, the Great Genetic Shuffle. We're going to break down how you end up as a totally unique mix of your parents, thanks to the elegant biological dance of meiosis I. But first, let's start with a question that seems simple but is actually pretty profound. You know, we all learn that we get half our DNA from each parent. So why don't we end up with double the amount of DNA? Why aren't our cells just packed to the gills with genetic code? I mean, think about it. If that happened every single generation, things would get way out of hand fast. It's not like our cells are playing some cosmic game of 2048, just doubling and doubling chromosomes until they hit a high score. No way. Life just couldn't work like that. Yeah, that kind of exponential growth would be a complete disaster. So your parents, they have two sets of chromosomes. We call that diploid, or 2N. If they just smashed all that DNA together, you'd have 4 sets, 4N. And your kids, they'd have a whopping 8N. It's just not sustainable. So clearly, life figured out a much, much smarter system.
So how does life pull it off? How does it solve this doubling problem? Well, the answer is it's elegant. It's a special kind of cell division, and it's called meiosis. This is the whole secret to keeping our genetics stable generation after generation.
So what's the big picture here? What's the main goal of meiosis? Well, if you think of your genes like a deck of cards, meiosis is all about one thing: having that deck perfectly before you pass it on. At its heart, meiosis is a special process for one reason. Its main job is to slash the chromosome number in half. It does this to create our reproductive cells, sperm and eggs. And this reduction, this halving, is the absolute key to everything. And here's how it all clicks into place. Most of the cells in your body are what we call diploid, or 2N, meaning they have 2 sets of chromosomes. Meiosis takes those cells and creates haploid gametes, sperm and eggs, with just 1 set, or 1N. So when a 1N sperm meets a 1N egg, boom, you're back to a stable 2N organism. Problem solved. Okay, but here's where it gets really interesting. Meiosis isn't just about cutting the chromosome count. It's also about shuffling the genetic deck. And the most critical part of that shuffle happens right at the beginning, in a stage called prophase 1. In prophase 1, something truly incredible happens. The chromosome you got from your mom literally finds its matching partner from your dad. We call these homologous chromosomes. They cozy up in a process called synapsis, forming this 4-part structure we call a tetrad. Each of those 4 parts is a chromatid, an identical copy of a chromosome. And this is where the magic happens. While they're all paired up, these chromosomes actually swap little pieces of themselves. It's called crossing over, and as the source material points out, it leads directly to genetic recombination. It's physically creating brand new combinations of your mom's and dad's genes on the same chromosome. This is a massive reason why you and your siblings aren't identical clones. All this shuffling and swapping perfectly sets the stage for the next big event, the Great Separation. And what's so cool is that these physical movements happening inside a tiny cell, well, they're the real-world basis for the famous laws of genetics that Gregor Mendel figured out over a century ago. Okay, so let's talk about Mendel's first law, the law of segregation. It sounds a little formal, but the idea is actually super simple. It just says that for any given trait, the 2 versions of a gene you have, we call them alleles, have to split up so that each sperm or egg only gets one of them. So where can you actually see this law in action? Right here, in a stage called anaphase 1. This is the moment the homologous pairs are literally ripped apart and pulled to opposite sides of the cell. The chromosome from your mom goes one way, the one from your dad goes the other. Mendel's abstract law is a physical reality. But that's not all. This whole idea of separation has a powerful partner, Mendel's second law, the law of independent assortment. This law basically says that how one pair of genes separates has absolutely no effect on how another pair of genes separates, as long as they're on different chromosomes. And the physical proof for this law? It's all about how those chromosome pairs line up in the middle of the cell back in metaphase 1. The way the long pair orients itself doesn't affect how the short pair does. It's completely random. It's like shuffling multiple decks of cards all at once, creating an astronomical number of new combinations of your parents' chromosomes. It's another huge engine for genetic diversity. So, after all that incredible shuffling and separating, where does that leave us? What's the actual outcome of all this work in meiosis 1? Here's the net result. We start with one diploid, 2N cell, and after this first major division, we end up with 2 cells that are now haploid, 1N. The number of chromosome sets has officially been cut in half. That's why we call meiosis I the reductional division. But hang on, notice the DNA content? We went from 4A down to 2A. Why not 1A? Well, it's because each chromosome in these 2 new cells is still duplicated. The chromosome number is halved, but the DNA is still waiting for one more split.
So let's just recap what Meiosis I managed to pull off. It's pretty amazing. 1, we've cut the chromosome number in half. 2, we've shuffled the genetic deck like crazy through both crossing over and independent assortment. 3, we've seen Mendel's laws play out in real time. And 4, we've created 2 completely genetically unique daughter cells. Not bad for one phase of division.
But hang on a second. Remember the definition at the start? It said meiosis creates 4 cells, and right now we've only got 2. Plus, each chromosome in these cells is still duplicated, holding on to its copy. So the deck has been shuffled and halved, yes, but the job's not quite done. How do we get from these 2 cells to the final 4 gametes? Well, that's a story for meiosis II. All right, welcome to The Explainer. Today, we're going to talk about something absolutely fundamental, something happening inside you right now. It's this incredible, perfectly timed dance that every single one of your cells is doing, all the time. Let's dive in.
So, let me ask you something kind of weird. What do you, a banana, and a fly all have in common? I know, it sounds like the setup to a bad joke, right? But the answer is actually fundamental to what makes us, well, us, to what makes us alive.
It's this, a perfectly controlled cell cycle. You can think of it as the universal operating system for life. It's the step-by-step process that lets cells grow, copy all their genetic info, and then split into 2. This is the engine that drives growth, helps you heal, and really, it's behind reproduction for pretty much everything on this planet.
Okay, so here's our game plan for today. We're going to look at a cell's life cycle in general, then we'll get into the details of that long prep phase, interphase. We'll also check out a special off-ramp called the G0 phase. And finally, we'll meet the all-important guardians of the cell, the checkpoints.
So let's start with the big picture. A cell's whole life is basically broken down into 2 main phases. And honestly, how much time it spends in each one, it's probably not what you'd expect. Yeah, see, you think a cell's life is all about that dramatic moment of splitting in 2, right? But nope, it actually spends a whopping 90% of its time just getting ready. That's called interphase. The actual division part, the M phase, is only about 10% of its life. It's kind of like studying for a final exam. You spend weeks and weeks preparing for just a couple of intense hours. That's a cell's life in a nutshell. Now, during that short, dramatic M phase, something really important happens. When the nucleus divides, it's incredibly precise. The cell's genetic blueprint, its DNA, has to be divided perfectly with 0 mistakes. But the rest of the cell, the cytoplasm, all the goo and organelles inside, that part doesn't always split evenly. And because that cytoplasm split isn't always 50-50, it means the 2 new daughter cells often start out a little bit smaller than the cell they came from. So they can't just turn around and divide again right away. They need some time to grow up first. And that brings us right to our next topic.
So what exactly is going on during that big growth period? Well, we're diving into interphase now. Remember, this is the 90% of the cell's life we were talking about. And it's anything but lazy. It's a super busy time of growth and careful preparation.
An interphase isn't just one big blob of time. It's actually broken down into 3 key stages: G1, S, and G2. And each one has a really critical job. First up, we've got the G1 phase, which stands for first gap. This is basically the cell's main growth spurt. Right after it's born, this little daughter cell gets to work. It's cranking out proteins, duplicating all its internal machinery, the organelles, and just bulking up to get back to full size. Then comes the S phase. S is for synthesis, and you could say this is the absolute heart of interphase. This is where the cell does its most important job. It makes a complete and perfect copy of every single strand of its DNA. It's a massive undertaking. And in animal cells, this is also when it copies the centrosomes, which are key for organizing the whole division process later on. And finally, we hit the G2 phase, or second gap. I like to think of this as the final pre-launch checklist. The cell's grown, the DNA is copied, now it just needs to make sure it has all the supplies for the big show. It's synthesizing key proteins, like tubulin, which it's going to need to build the ropes that pull the chromosomes apart. It's just a final check to make sure everything's ready to go. Now, it sounds like the cycle of growing and dividing is what all cells do, but it's not. Some cells actually take an exit.
They get off this highway of replication for good. This exit ramp leads to what we call the G0 phase. It's basically a resting state. Think of it like cellular retirement. The cell has officially left the cycle. It's still alive and doing its very specialized job, but it's just not planning to divide ever again. So which cells do this? Well, some of the most famous examples in your own body are your nerve cells, your neurons, and your muscle cells. Once these cells mature, that's pretty much it. They stop dividing and just focus on doing their specific, important job for the rest of your life. Okay, so this whole process is, as you can tell, incredibly complicated. And just one tiny mistake, a little bit of damaged DNA, an incomplete copy, could be a total disaster for the organism. So how does the cell prevent that? Well, this is where the heroes of our story show up, the guardians of the cell cycle. So, yeah, how does the cell manage this amazing level of quality control? How does it stop itself from making mistakes that could lead to all sorts of problems? The answer is a series of internal checkpoints. Think of them like security guards. There are 3 main checkpoints that are absolutely critical. We've got the G1 checkpoint, the G2 checkpoint, and the M checkpoint. Each one is like a strict security gate the cell has to get permission to pass through. Let's start with the first one, the G1 checkpoint. You could argue this is the most important one of all. It's the bouncer at the door of the S phase, and it's asking some really tough questions. Hey, are you big enough? Do you have enough resources? And most importantly, is your DNA okay? Is it damaged? This checkpoint is a really big deal. It's basically the point of no return. If the cell passes this check, it's all in. It's committed to dividing. But if it fails, if the DNA is damaged or something's wrong, the whole cycle hits the brakes. The cell will try to fix the problem. And if it can't be fixed, the cell gets the order to, well, to self-destruct. It's a safety mechanism to protect the whole organism. Okay, so let's say the cell passed G1 and finished copying its DNA in the S phase. Now it hits the second security guard, the G2 checkpoint. This one is like a super obsessive proofreader. It's poring over the new DNA copies and asking, okay, is this copy job actually done? And are these copies perfect? No typos? And this is so important, because this checkpoint stops the cell from dividing if the DNA copies aren't right. It is literally the last chance the cell has to fix any mistakes before it splits and locks those mistakes into 2 new cells forever. All right, our last guardian shows up right in the middle of the action during the M phase. The cell is all lined up, ready to pull its chromosomes apart, and then halt. The M checkpoint steps in for one final check, and it's asking, is every single chromosome hooked up correctly? Are they all lined up perfectly in the middle? This check is all about preventing one specific disaster, giving the wrong number of chromosomes to the new cells. If even one chromosome isn't attached right, the daughter cells would end up with the wrong amount of genetic material. That's a recipe for serious problems, like genetic disorders or even cancer. So the M checkpoint literally pauses the entire process until everything is absolutely 100% perfectly in place. So when you look at the whole thing, from that long, careful preparation and interphase to the watchful eyes of these checkpoint guardians, the cell cycle is just an incredible masterpiece of control. It's how life manages to go on with such stunning accuracy. Which of course leaves us with a really big and kind of scary question. What happens when all that control is lost?
All right, welcome to The Explainer. Today we're diving into a tale of 2 very different cells, both of which are absolutely critical for human life. We're talking about the sperm and the egg.
You know, this single question really gets to the very heart of our story today. And by the end of this explainer, you're going to understand the completely different life stories of these 2 cells, and you'll see exactly how those differences solve this mystery.
So to really get a grip on their journeys, we have to go way back to the very beginning. And what's so fascinating is that the first major plot twist, it happens right out of the gate.
Okay, get this. This is the first huge difference. For males, the whole sperm production factory doesn't even open for business until puberty. But for females, their entire lifetime supply of potential eggs is created and stored before they're even born. Wild, right? Now, before we go any further, there's a key term we should probably nail down. Both sperm and eggs are specialized cells we call gametes. Really, they have one job and one job only, to carry half of the genetic blueprint, with the goal of fusing together to create a brand new individual.
So let's look at the production process itself.
because this is where the two strategies just completely go their separate ways. One is like a non-stop marathon, while the other is this decades-long journey filled with these really critical pauses.
For males, the process is called spermatogenesis, and you should really think of it like a production line. Starting at puberty, you have this lifelong reserve of stem cells that just continuously enters the pipeline. After a couple rounds of division, each of those starter cells results in 4 mature mobile sperm. It's just a super efficient marathon that runs for decades.
For females, though, it is a completely different story. It's really a story of suspended animation. All the potential eggs get formed way back in the fetal stage, and then immediately they hit pause. They're just arrested in the middle of their first division. Then, from puberty on, one of these cells gets chosen each month to resume its journey, only to pause again right before it's released. It will only, and I mean only, complete its mission and finish dividing if it's successfully fertilized.
And these different strategies, they become even more dramatic when we look at the final numbers. It really is a story of overwhelming force versus incredible selectivity. I mean, this difference in scale is it's staggering. The male strategy is all about quantity, pumping out hundreds of millions of new sperm every single day. The female strategy, on the other hand, is all about quality, investing all of its resources to mature and release just one single egg each month. So that kind of begs the question, right? Why this huge difference in the numbers? Why make millions and millions of tiny cells instead of just a few big ones? Well, the answer is all about how they divide a resource that's just as vital as DNA, the cytoplasm. And this is where the different game plans just become crystal clear. In sperm production, the cell's resources, its cytoplasm, are divided up equally. You get 4 small, streamlined racers. But in egg production, oh no. The division is brutally unequal. It's a winner-take-all scenario. One cell, the future egg, basically hoards nearly all the cytoplasm and nutrients. The other 3, these little things called polar bodies, they're left with almost nothing and just fade away. They're a sacrifice to make sure that one egg has absolutely everything it needs to start a new life. Okay, I know this table looks a little technical, but the core idea behind it is actually pretty simple. Both sperm and egg cells need to cut their genetic material in half. That's the whole goal. They start with a full 2 sets of chromosomes. Then they go through 2 separate rounds of division, carefully whittling down that genetic material until each of them is holding just a single set. And this is so important because it ensures that when they combine, the resulting embryo gets the correct, complete amount of DNA, 1 set from each parent. So, why does all this stuff matter? Well, because these biological details have these profound, real-world consequences. And they bring us right back to the mystery we started with.
And here it is. Here's the answer to our mystery. The biological clock is a direct consequence of that egg's decades-long pause. Think about it. Sperm are constantly being made fresh, but an egg that's released at age 40 has literally been held in suspended animation for 40 years. That long wait increases the chance that when division finally kicks back in, the chromosomes might not separate perfectly. This directly explains why the risk of certain chromosomal issues increases as the mother gets older.
And for sperm, there's another really fascinating detail. Their development requires this very specific temperature, a few degrees cooler than the rest of the body. This one single factor is the entire reason the testes are located outside the body cavity. It's basically a dedicated climate-controlled environment just for mass production.
So in the end, it really all comes down to 2 brilliantly different solutions to the exact same biological problem. You've got a numbers game with millions of tiny racers on one side versus a high-stakes investment in one well-provisioned champion on the other. Both are just remarkable strategies perfected by evolution to work together.
It's really an incredible example of biology's diverse solutions, which leaves us with a final question to think about. The nonstop factory or the patient, decades-long wait. Which strategy do you find more remarkable? Thanks for watching.
Hey everyone, and welcome. Today, we are diving deep into one of the most incredible, fundamental processes of life. It's this tiny, microscopic ballet that lets every living thing, including you, grow and heal. It's called mitosis. And yeah, it's happening inside you millions of times as we speak.
I mean, have you ever really thought about that? How does one single cell eventually become, well, you? Or how does a simple onion bulb sprout into a full plant? The answer to how a scraped knee heals or how a giant tree grows is, believe it or not, the exact same process.
The secret is mitosis. It's this beautiful, super precise dance of division that's constantly happening in our cells. So come on, let's shrink way, way down and get a front row seat to the show.
Okay, but before we watch the performance, we gotta know the rules. You see, biology has a very specific way of keeping score when it comes to our genetic material. Once you get this part, everything else just clicks into place, I promise.
So first up is this idea called ploidy, which we just use the letter N for. The easiest way to think about it, imagine one N, one complete set of chromosomes, is like a full combo meal. You know, you get the main thing, the side, the drink. It's the whole package, genetically speaking.
Right, next up, we've got the DNA amount, which we label with an A. Now, if N is the combo meal itself, then A is the actual amount of stuff in that meal. It's the raw quantity of genetic material, the actual DNA, in one of those complete sets.
So here's the deal. Most of the cells in your body are what we call diploid. That means they have two of those combo meals. So they have two sets of chromosomes and naturally two corresponding amounts of DNA. That gives us our starting formula, 2N, 2A. Burn that into your brain, it's our starting line.
And this right here is basically the entire roadmap for our journey. We start at 2N, 2A. The cell then gets ready to divide by copying all its DNA, which takes it to 2N, 4A. Then comes the big moment, a really brief, dramatic peak where everything doubles to 4N, 4A, right before it splits into two brand new, identical cells, each one back at that stable 2N, 2A state.
Now listen up, because this is the part that trips almost everyone up. When the cell copies its DNA, it doesn't create new sets of chromosomes. The number of meals, our N, stays the same. It just doubles the amount of stuff in each meal. So you still have two combo meals, but now they've both got double patties and you've upsized the fries and drink. The amount of DNA, our A, has doubled to 4. All right, you with me? Now that we've got the rules down, let's watch the dance itself. Mitosis is really one continuous flow, but to make sense of it, we break down the choreography into 4 main acts. The performance kicks off with prophase. The DNA coils up super tight into these thick, visible threads. Next, in metaphase, these chromosomes line up right at the cell's equator, ready for action. Then, the climax, anaphase. The copies are yanked apart, and that's our temporary spike to 4N, 4A. And finally, telophase, the grand finale, where 2 new cells take shape, each getting its own complete set of instructions, bringing us right back to 2N, 2A. Let's just pause on that one moment for a sec, because it's so important. Before anaphase, you have these X-shaped structures. Each one is a chromosome with its identical copy attached. We count that as 1 chromosome, so the cell is 2N. But the very instant those copies are pulled apart in anaphase, each copy is now considered its own chromosome. And just like that, for a brief moment, the cell has double the chromosomes, a sudden wild jump to 4N. Okay, enough theory, enough diagrams. Let's see what this looks like for real. We are heading to one of the absolute best places in nature to see mitosis with our own eyes, the tip of an onion root. And to see it, we need to get really close. We're talking 400 times magnification. At that scale, the hidden world inside a single cell just pops into view, and we can actually watch this dance of division happen. So if you actually look at a slide of an onion root, you might notice something surprising. Most of the cells you see are, well, kind of just sitting there. They're not dividing, they're in what's called interphase. And that makes total sense, because the prep work, all the growing and copying of DNA, takes up about 90% of the cell's entire life. The actual division part is a quick, final sprint. Think of this table as our spotter's guide. If you see what looks like a bunch of dark, scattered threads where the nucleus used to be, boom, that's prophase. When those threads get organized into a messy line down the middle, that's metaphase. And then the money shot,
If you see distinct V-shapes being pulled to opposite ends of the cell, that's anaphase. That's our 4N4A peak. And finally, when you can see two new nuclei forming with a faint line appearing between them, that's telophase. The split is almost complete.
Oh, I just love this description. That V-shape you see in anaphase is so clear and dynamic. It's not like they're gently floating apart. No, they are being pulled. You can almost feel the tension as these little fibers reel them in. It's a tug of war, and that V-shape is the result. It really is the climax of the whole dance.
And that faint line we see in telophase, what's that about? Well, because plant cells have a tough, rigid wall, they can't just pinch in the middle like an animal cell would. So instead, they build a new wall from the inside out. All these tiny bubbles filled with wall material line up in the middle and fuse together, forming something called a cell plate. It starts faint and then grows until, poof, a new wall separates two new daughter cells.
So, we've covered the rules, we've walked through the steps, and we've even seen the evidence under the microscope. But so what? Why does this tiny, intricate dance actually matter? Let's zoom back out and connect the dots. One really cool indicator of a major growth zone is something called the nucleus to cytoplasm ratio. See, when a cell divides, the two new cells are small, right? But their nuclei are already full size. This means there's a really high ratio of nucleus to everything else in the cell. So when you see an area packed with these high ratio cells, like an onion root tip, you know you found a hotspot of furious growth.
When you step back, it's incredible. This one single process explains so much. It's how we all grew from a single fertilized egg. It's how your skin heals after a cut. It's the engine behind any organism that reproduces on its own. And it's why a humble little onion root gives us this perfect, beautiful window into the mechanics of life itself.
Every single step of this cellular dance is perfectly timed, perfectly regulated, checked, and double-checked by the cell. It's a flawless system perfected over billions of years of evolution. And that leaves us with one last, really critical question to think about. If this perfect dance is the key to healthy life, what happens when a cell forgets the steps?